This is the April 22nd meeting of our IMAG working group on multi-scale modeling and viral pandemics. We have two exciting mini seminars, and as usual, I think 20 minutes is not going to do justice to either topic, but both speakers have generously agreed to stay on for the half-hour open discussion at the end. So that'll give us time to explore a bit more. And as usual, I'll say the same thing, which is we hope that you'll come back and give a follow-on talk and explore more of your work another time. As always, I have to remind people that this meeting is currently being live streamed on YouTube, that it's being recorded, and that those recordings will be made publicly available. As usual, we have Reinhard Labenbacher and myself, James Glazier, as co-leads, and the excellent support, both scientific and administrative, from Jim Sluka and Bruce Shapiro. You have our email addresses. If there are any issues, comments, suggestions, questions, complaints, please feel free to contact us. We have the usual slide that you've seen many times, that we have a Slack channel, a Twitter account, an IMAG MSM wiki page, a LinkedIn page, a YouTube. We also have a Facebook page. If we use them, they're helpful. If we don't use them, they're less helpful. Please help us make those social media connections work for everybody. I particularly encourage people to point to the archive, which is growing rapidly, of wonderful YouTube videos recording all of these talks. I think it's a great resource, and I hope people will take advantage of it. And now we have time for announcements. Are there any short announcements that people would like to make about subgroup meetings, about funding opportunities, conferences, anything that you'd like? I don't see
the innate
any.
immune response subgroup is meeting on Monday at 4.
Any other subgroup announcements for timing? Anything else that we need to know about? Feel free to, you don't have to raise your hand, you can speak up if you like.
Dissemination and outreach will meet on next Wednesday at 9:30 for the whole group. If you're interested in not receiving the links, let me know and we'll add you to the observer list.
Reinhard, is there a crowdsourcing meeting tomorrow morning, or is that canceled?
No, there is tomorrow morning at 11, I believe.
Any other subgroup meetings, a conference? No? Okay.
I'm sorry,
So,
10 o'clock.
So next week we have Penelope Morell speaking. She's an immunologist. I hope that will be a very interesting talk. I've heard her speak before. We have Catherine Morse, who's going to be talking about standards and standard development. You'll hear the best practice, not just in our little world, but in the big world. That will be exciting. The following week, we have two speakers booked. May 13th, Fred Adler has very generously agreed to do the subgroup presentation. We encourage people to do more of those, and John Rice will also be doing a subgroup presentation. As always, suggestions for future speakers and material are appreciated. Please mute yourself if you're not speaking to help reduce background noise. Please hold the questions until we've gotten through both talks so that we have as much time as possible for our speakers. Both speakers this week have generously agreed to do the 30-minute after meeting discussion. I hope many of you will be able to stay on and participate. And I will give a 5-minute warning for our speakers. So if I break in and say 5 minutes, please don't be offended. It's such a tight schedule to try to get two talks into an hour with a little less that it's difficult. So without further ado, people know that we don't do big formal introductions for this meeting, but I'm very pleased that Debra Peters has agreed to speak to us. I heard her speak at NSF a few weeks ago and was really blown away by the combination of experimental and modeling work that she did, and also on her work on how to organize and conceptualize large projects. And she presented so much material then, there's no way she could present all of her work in 20 minutes, but I hope we will get a flavor of her exciting approaches. So with that, I will stop the share, turn it over to you, Debra, and thank you very much for agreeing to speak.
Great. Okay. Thank you. Let me get my. You see my screen, or do I need to start over? I think I tried. There we go. Okay. Got it here. So are you seeing my first slide or somewhere down below?
We're seeing your PowerPoint presentation, not the display mode, but the desktop, your author view.
Okay, all right. There we go.
There we go. Perfect.
Okay, well, thank you for inviting me. I'm going to give you sort of, as he said, as James said, a flavor of my work, basically integrating lots of data with modeling in a multi-scale approach. And in this case, to predict the spread of vector-borne diseases. And actually, what we'll do today is to focus on vesicular stomatitis. We see that as a model disease that we can use for many other vector-borne diseases. And then our goal is then to develop a strategy and framework that we can use to actually integrate all this information in a predictive way. And if we have time, I'll go into another example that has to do with the Dust Bowl to show you how this approach works for other totally different. I'm really interested in catastrophes and things that sort of start small and get out of hand in a hurry in space and time. So here's my website if you're interested in that. Okay, so vesicular stomatitis, we'll just go with VS from now on. It actually, what's really interesting to me is that it has multiple hosts. We're going to talk about just horses today because that's where most of the data are. There are two serotypes. Since about 1988 or so to 2018, it was New Jersey, the United States, and then 2019 and 2020 threw a sort of a wrench in it and became Indiana. So we don't really know a lot about the two serotypes. We do have two serotypes, and then multiple insect vectors. And I'll talk about black flies and biting midges. What's really important about this is it's actually a fairly minor, it's one of the largest ones, but it doesn't actually create mortality or anything. But the issue is that it looks like foot and mouth disease. So that's why the US is so concerned about it. And they need to rapidly diagnose which one of those it is. And we haven't had foot and mouth in the US since 1926, but they're really worried, the USDA is, that if that happens again. It's a reportable disease then to the USDA Animal and Plant Health Inspection Service. It has sort of a 6 to 10 year interval in the past, although we just had another one, another outbreak in 2019. So now it seems like potentially it's more frequent. Well, I was at a conference and I was in the audience and I heard Luis Rodriguez from the Foreign Animal Disease Lab from the USDA give a talk about this. And I'm a landscape ecologist, systems ecologist, so I don't know anything about diseases. But he said, we don't understand this because sometimes in 1 year, like 2004, the points are VS, it'll go to Colorado. The next year, 2005, it'll go all the way to Montana. He said, we have no idea what are the drivers are of these two different kinds of patterns. So in B here, that's all of the points. And so the first thing I did was separate it out into the incursion year and then the expansion year. But this was the initial problem. So how do you explain this sort of broad scale pattern? And this is a heat map. So it starts, in the south, Mexico is the endemic region, and then it spreads to the north. And that's just the pattern. So this is our first attempt. How do we actually explain this? So Luis and I knew that we didn't have the expertise to do it all. So we brought in the insect vector people from Kansas, a range scientist from Colorado, and an economist. You have to actually put the animals into the environment. Then our APHIS colleagues, they have all the data, right? We have now people from Mexico State University working on the endemic region. And then in my group, I have an eco-hydrologist because the part of black fly population dynamics are spent in flowing water. So we needed somebody who actually understood water. And then the rest of my people, they're my staff, and they're actually just really talented in data wrangling. They're data scientists, programming, machine learning. So we're sort of the glue, and I'm the integrator, right? So you got to have somebody who actually can take all these pieces and put them together. So our next step was, I asked Luis, where's your systems diagram? And he said, well, he didn't even know what one was, right? So the first, to me, that's what you had to do, is to actually put together, how do all these different components fit together? So the insect population dynamics, then how do they actually infect the hosts? The host can then, infect other insects. And then that's sort of a local scale environmental conditions. And then how do they spread? Right, so that's insect dispersal. Sometimes the horses actually get transported, by vehicles, and our APHIS people know all that, so they have the knowledge about that. But you have to actually then, take these dynamics from like what we call premises, which are ranches, and then get it to spread across the country, right? So that's sort of the landscape scale processes that are also need to be accounted for. So what I didn't want to do was to just do correlation analyses. What you see a lot in the literature for these diseases is that they'll just look at climate, right? Precipitation and temperature, and just see if it's correlated with the patterns that you see. I wanted to take a process-based approach. So on the yellow boxes on the left, those are all the processes from the previous slide. And then we ran through the different driving variables. For example, this one in green is soils. But it's like, what is it about soils, right? So soils is our driver, but the actual variables, it could be more than one, are what explain the process. So we start with, a lot of times we have information from the lab on like number of eggs produced and water temperature or water depth. That's from the lab, but now we have to get it into the field, right? We know our patterns, and so the main soil characteristic is available water holding capacity, and that affects the depth of water, right? So then we can actually say, okay, now it's available, it's AWC is our explanatory variable, and here's the relationship that we expect to see with the density of the eggs. So I think that's what we were trying to get at, and that's the difference to me between drivers and variables. So climate is a driver, but the variable is, seasonal precipitation. And then we need the relationship between that variable and each one of these processes. So I think this is what's critical to how our approach sort of differs from a lot of other people. And then when you do that, you have to figure out the data, right? So now you know your variables, and you have to go out and actually find all these data. And this is where my people came in terms of the drivers on the left, and then the actual variable is the fourth column, I guess, or fifth column. So this was the key, but then once you have all the data, which it's very easy to download. All of the data here are open online. They're standardized within the data set. So NOAA standardizes all of their climate data, but they don't standardize with the NASA imagery, right? So we then had to actually, what I call harmonize, putting the data together in the same spatial resolution and temporal resolution. And that really is that's quite a challenge. But our grid size, and we did a continuous grid across the Western United States, was 4 kilometers by 4 kilometers. And that's based upon the response data. So what APHIS does is they place the occurrence at the mailbox. Regardless of how large the ranch is, that's the location of where the horse got bit. And so we then said, okay, well, based upon ranch sizes, our grid size should be about 4 kilometers by 4 kilometers. So we harmonized all the data at that grid size. And then we conducted the analyses. In this case, we used its maximum entropy model. It's a machine learning model that takes all of this information. We have almost 500 layers of data that we had to put together across the entire Western US, and then run the machine learning analysis. And so this is just a view of some of those results. The interesting thing is that if you look at the bars on the left, that's the incursion year, 2004, expansion year, 2005. You need all of those drivers, right? It isn't just climate, right? You actually need soils in 2005. You need distance to water. So that distance to water is a habitat for the black flies. And then soils, AWC, is an indicator of the habitat for biting midges. So now we actually have a hypothesis that that black flies are the vector in the first year of an outbreak based upon our habitat variables that came up, and biting midges become important in the second year. And so I see these as hypotheses, because when we started this, Luis said, we don't know the environmental variables, and we're not even sure about the vectors. So these are hypotheses that we're now testing. So the insect people are going out and actually testing and looking for black fly abundance. Is the virus in the black flies? So this has now led to a whole nother scope of work in terms of figuring out, but we have this to guide us, right? So I think that was a critical thing for our work. And the interesting thing on the right is that 2004 habitat variables or environmental variables are the same in 2014, and 2005 are the same as in 2015. So we have the viral genetics, right, to say that 2004 is the same outbreak as 2005, and 2014 is the same. The virus is the same in 14 and 15, but it's driven by the environmental variables. So this was totally enlightening to everybody, and totally unexpected that this would happen. We actually thought, 2004 and 5 would be related environmentally, and 14 and 15, but it actually turned out to be quite different. So that's the gist of the sort of VSV work. We're expanding that now to West Nile, looking at the EEE data. It really depends upon the response data that we have available. And so those APHIS has a lot of information online. So we're expanding to other diseases. I wanted to give you a flavor of sort of how this works with a totally different kind of problem, because one of the things I was asked to talk about was sort of opportunities for collaboration. So here's just something interesting to think about, right? So here's a map on the left, and if you've ever looked at Dust Bowl literature or read anything about it, right, was the massively devastating consequence in the 1930s of the drought across much of the Western US. And then combined with farming practices, we had these huge dust storms and losses of cultivated fields and everything. So on the left, you see the map, and you see this all over the place. It's the same map. And the red is sort of where the most intense part was, but then the yellow is the extent of the Dust Bowl region. And it's like, and I was looking at this like, so why did it stop in Iowa? So I grew up in Iowa. So I always have this sort of thing when I see Iowa, right? Well, there's a boundary between Nebraska and Iowa. What was the reason for that boundary? And so that's really what led to this project, was like, is that boundary real, right? How did they draw it there? And then what's the cause, right? What are the environmental variables? And so we use the same approach as we did with the VSV. So what you're seeing in the colored box is ABC. That's the boundary. So Nebraska's on the left, Iowa's on the right. The Missouri River runs between the two. And so we just focused on that boundary. And so then we actually went in, and another USDA agency, the National Agricultural Statistics Service, has a lot of county level data going back. So we had corn from this time period, the Dust Bowl, the pre-Dust Bowl, and the post-Dust Bowl. And so that's what's shown on the bottom there is, okay, if you had driven from Nebraska to Iowa, you would have seen a marked difference in production. So the blue circles are production in Nebraska, and the green ones are production in Iowa. There's a complete change, right, to go across that border, just for these little counties. And so it's not precipitation, it's not temperature. The first thing we looked at was soils. It's not any of that, because Iowa and Nebraska, they overlap in all of our sort of traditional variables. This was quite perplexing for a large number of weeks. I run with my dog, and at some point during a run, I said, it's the river. So the only explanation that we have, and it's a hypothesis, but that so I got this map off USGS from the 1950s. So you can see sort of I think I have to grab but you can see that the floodplain of the Missouri River was actually really large. It's a 10K, right? It's 6.2 miles in places, and that's hard packed. So the river at that point in time was this crazy wild thing. It just went back and forth throughout the floodplain, right? The alluvial plain. It's totally different now because it's been managed since the 50s. But in the 1930s, it just didn't. It ran all the way back between there. And if you actually look, it's really, you can see that the river is like a little blue line that runs through there. So, but it could get really big, right? But what kept it in the floodplain is that when you come across Nebraska, you go down an elevation to the floodplain, and you go across the floodplain, and you have to go up into Iowa again. It's another elevation. So what was happening? So if you look at the blue maps on the bottom show you the abandoned fields. So Nebraska had lots of abandoned fields. Iowa, no, right? So, okay, we've verified that, in Iowa, the tremendous devastation happening in Nebraska was not happening. The corn was just decreasing in production, but it was not getting sort of covered by sand. And so I think what we then did is went in and did a tremendous literature search. And all the dots in the map on the right are all places, the purple ones are places that showed dust particles in the air. So the monthly weather review, I guess, is the one that actually documents these things. So all of those places had actually dust particles. So the Dust Bowl is expanding all the way to the East Coast. The red and the blue are actually points where people were sampling, right? And then the green is in Iowa. So again, on native prairie, not just the corn, the native prairie, the green dots, again, all that the plants did was decrease in size. In red, the plants all died. So another confirmation of the boundary. But I think what we're suggesting here is that the boundary is associated with the river. So it's this landscape scale sort of feature that's not typically considered, at least not for ecologists, that you could have a river actually stop the sand moving across the landscape. So the photos there show you that, these huge dust storms, they would actually have sand creep right along the ground and then cover all the fields and kill them, right? And so when they got to the Nebraska border, they had to go down an elevation, the sand would have been deposited. And then they had a whole 10K of area that had no sand source. So Iowa was totally protected, we think, from this sort of devastation because of the Missouri River.
Sorry, 5 minutes, if it's okay.
Okay, good. Yeah, so I'm actually wrapping up. So the insights are, I think a transdisciplinary team is needed, and someone to actually be the glue, right? You can't have just the pieces. It's an iterative process from data to metadata to scientific expertise, and then back to the data again. And we're generating hypotheses, right? And focusing on these pattern process relationships. And then we have to be adaptive because when the next outbreak came in 2019 and 2020, it went all the way into Missouri. So like totally different location that we have to now deal with. So our challenges and opportunities, I think, for collaboration is the data availability. We don't know a lot about insect vectors' response to the environment. These two serotypes, we still know absolutely nothing about them. They seem to have different habitat conditions, and the use of multiple computational tools, and then really interested in these sort of cross-disciplinary comparisons with these similar dynamics in a search for generalities. And then I just have a slide of the papers for posting so you can get to those. Okay, thank you.
Okay, thank you very much. That was a wonderful talk. We'll hold the questions, and since they're so short, we'll hold the questions until we get through both talks, and then we'll have plenty of time for discussion. I think there'll be a lot of questions about organization and project structuring too. That's wonderful. Thank you again. So, we'll move on then to our second speaker, Donald Combs from Eastern Virginia Medical School, talking about the Digital Patient Project. So now we're going to move from very large scale to individual modeling. And I'd like to, are you ready to take it away, please?
I am. I want to make sure I can share the screen. Do you see my slides?
Not yet. Just give it a second. And again, if it's okay, I'll break in at the 5 minutes mark.
Yeah. So I've shared the screen, I thought.
There you go.
Okay. Well, thank you very much for the invitation to speak. And it's absolutely appropriate to follow Debra Peters' presentation in the sense that her emphasis on how you organize a project. I'm sorry.
Before that, you're not in presentation mode. If you want to be.
No, I want it that way. I can track on the left where I am.
Okay, that's fine. Sorry, I
Yeah.
just wanted to make sure I apologize. Please start.
It was
Sorry.
actually intentional.
Okay,
Not
thank you.
everything I do on the computer is. At any rate, I think her emphasis on the importance of organization is really important, because this is not so much a brand new project or a startup, but a reinvigoration of an existing project. And I want to spend a little bit of time trying to do some definitions. But the image that I want to start with, and I think almost everybody will be familiar with it, is Charles Dickens' Christmas Story, where the character Scrooge is visited by three ghosts, the ghost of Christmas past, present, and future. And you can see, in letting the story unfold, the factors that led to him being the way that he is, and if he doesn't change, the differential outcomes in the future. And I think that's what we hope for ultimately with the digital patient, is to be able to understand the factors that lead to a current status of health and understand the implications of different approaches thereafter. So let me get going here. So I think it's important to start with the big goal, and the big goal I think is pretty well captured in the by the Institute of Medicine when they talk about what do we want from healthcare. Well, we want it to be safe, we want it to be effective, we want it to be relevant to me, we want it to be timely, we want it to be efficient, we want it to be equitable. And all of those variables as a measure of the quality of healthcare are important. And this is where the digital patient, once realized in some number of decades, could help address these issues, I think, very carefully. This focus on the digital patient is part of a regional effort in Eastern Virginia that involves developing a platform and applications, as I'm going to talk about with the digital patient, but also assembling a huge amount of data from healthcare systems, from the Census Bureau, from the cities, from individual practitioners, from patient histories, into what these days used to be called a data warehouse, but now I guess it's a data lake. And so we're in the process of creating such a thing regionally with huge amounts of different sorts of data. And the purpose is then, can we take an application, which the digital patient is, and draw on all those sorts of data to build something that is useful. So the effort that I'm going to describe, or the thinking coming forward, is built on the efforts of lots and lots of people. I edited a book on this about 6 years ago, trying to pull together the work literally of hundreds of individual scientists around the world. And then there's kind of been a pause because it's such a big project. So it's important. And if we can build it, it will really, really, really make a difference in improving healthcare. So what is a digital patient? It's the latest in a long line of development. Back in the 1990s, the International Union of Physiologists started, well, actually before that, even the United States started. Remember the male and female that were sliced and diced anatomically, called the visible human? That was an effort to begin to build a model of the structure that was far more sophisticated than we'd ever had. That was a late 80s project. In the early 90s, the Physiome project, Peter Hunter down in Auckland, among others, headed that up to build a physiological model of the human body, all of the processes. So if the anatomy gives us the structure, the Physiome gives us the processes. In the early 2000s, the European Union funded a project called the Discipulus, the virtual human physiome, trying to go beyond the modeling that was traditionally done and is still done in physiology. And they've spent about 5 years, stopped around 2015. The funding stopped, and it morphed for a little while into something called Avicenna. But it's been pretty much stillborn. And so what we're talking about is an effort to reinvigorate the notion of what we're now calling the digital patient. So what is a digital patient? And this becomes really important. Taking one of the European Union workers on the virtual human project, Vanessa Diaz Zuccarini, defines it as a technical framework that once fully developed will make it possible to create a computer representation of the health status of each citizen that is descriptive, interpretive, integrative, and predictive. That's a really good definition if you draw a circle around the word framework. That's what we're focusing on. There are lots of people working on different models in silico, viral models. Dr. Glazier wrote an article with some colleagues not long ago about the notion of digital twins. There's a lot of discussion about models. In fact, this group is a part of an interagency modeling group at a federal level. There are lots of related concepts to this framework. Artificial intelligence, I already talked about data lakes. You can pick up journals every day and read about things like organs on chips and artificial organs and in silico R&D, different taxonomies for what the pieces are, how they fit together in ontologies. The point being that there is an interest in the big picture, which is what I'm interested in, the digital patient. There's been a lot more interest in people building specific, generally physiological models of heart-lung interaction, of kidney interaction, of the process by groups like the Human group at Mississippi or the Gears group that are trying to build, and all kinds of in silico models in pharma that are trying to build pieces. And so, for example, the digital twin, to me, is a subset of a digital patient because it's more narrowly focused. The notion of a twin is it's a twin to something that exists. It's only virtual rather than the real thing. The digital patient, I'm envisioning really as a platform where lots of things can go together. And so I think this slide kind of captures it of the digital patient's right in the middle, but it's built, ultimately, it is not built today, and it's going to be quite a while before it is, will be built on data sources ranging from the molecular to the neighborhood. And that's a major multi-level modeling challenge because not only is it multi-scale from the molecular to the social, because we know, for example, obesity is related to family and friends and neighborhood customs and culture and behaviors. And we've got to get that sort of data. We can't just stay inside the human body, but even if we stay in the human body, the scale of time varies greatly from what's going on at a molecular level to a tissue level. We have to make all of that somehow fit together. So what we are planning to focus on here is not doing so much of the individual work, although we want to test it, is we want to try to build an open source platform that people can plug in all sorts of data that over decades will grow into the capacity to do a digital patient, where they might, for example, take my actual data on, they might do a variety of lab studies and X-ray type studies of my cardiovascular system, but then they might not have as good data on, say, kidney function. And the idea would be you could take some of my personal data and take normative data from a normal kidney and plug it in and at least run that model going forward so that you have some actual data and some normative data. But in order to do that, you have to build an open source platform where people can drop in different pieces of the data and run analyses. And that's what we're talking about here. Just not to spend any time, but this is the range of issues that we have to deal with, everything from structured data to semi-structured data to somebody's verbal recollections, and how do you pull all that together to get the information that you need. These are issues that we're going to have to address. So I hope in a very quick period of time, without the sort of specificity you would give in an article, I've outlined the big picture of what it is we hope to do with our digital patient project, and that is to build a platform that people can take existing models and existing data developed for different purposes, some of it real time, some of it historic, so that we can move toward this notion of getting a model that really allows us to understand history, understand more fully what's going on today, and project alternative futures based on interventions. I've chosen to highlight just a few challenges. There are undoubtedly hundreds of them, but one of them is making a user interface that makes it relatively easy for people to import data and run analyses. And in that platform, it has the capacity to pull together data on different organ systems and plug in the missing pieces to calibrate between different space and time scales. That's a huge challenge, obviously. Another challenge is, and it's interesting that the EU effort wasn't funded on the healthcare side of their funding, it was funded on the ICT side, because they understood the biggest barrier is getting a platform where people can, I don't know, it's not a great analogy, but like build a Lego castle. You can put pieces together in different ways to come up with a different result that is coherent. Providing, we would think that in 2021, there is a common definition of a knee and what connects to the knee. But in fact, there are subtle variations. Some models include almost everything. Other models include less. And one of the keys to this effort is to work with those agencies that have been working on these issues for a long time, but to get an adoption of a commonly understood taxonomy and ontology among all of these types of data that have to come together to build a digital patient. When you start talking about massive data and repositories and who's going to own it and who's going to own access, it raises a whole bunch of issues. And we have to begin to at least, again, set a framework of how we're going to talk about that. In today's paper was a lengthy story about the new regulations on the use of artificial intelligence issued by the EU Commission just yesterday. So there's a lot of attention, and it's understandable. If we get this kind of data about individuals that we've never had before, the possible abuses are enormous. And so we've really got to pay attention to that in design. Another challenge is, I can go, this is kind of the framework. The challenge is that we need to build a group of people that are interested in focusing on the digital patient platform. It's the kind of commons problem that was written about many decades ago. In order to really advance in silico medicine, in order to look at quick, rapid vaccine development, for example, in the COVID-19 pandemic, there's a lot of interest in being able to do things in silico very quickly. But that will involve collaboration across research programs, and it will involve people not only paying attention to the particular issue that they're investigating, but to the broader environment and how that piece may fit into that broader environment. So my point being is this is not going to be done in Hampton Roads, Virginia. It's not going to be done in Virginia or in the United States. We've got to figure out a way to get points of view from all around the world. I don't mean every country has to have a representative, but we need enough diversity of points of view, thinking about how do we build the commons, what are the constraints, what are the capabilities. And I'm hoping that we'll spend the next year convening people to address that issue. As Debra Peters showed in her slide, it really helps to have a conceptual model, an end goal of what it is you're trying to address. And this is such a big issue that we need to spend some time trying to get the definition of what it will do and won't do. That's really a slide that just says, in order to realize a digital patient, we're going to have to be open to new knowledge. I think all of us that have lived through this COVID-19 pandemic have perhaps been struck by the importance of the word novel I know I didn't appreciate the full dimensionality of what a novel virus really means. It means we don't know that much about it. And so all of a sudden we're getting what, a third of the cases that are spread by asymptomatic people. And we're getting some people that are indicating respiratory distress, but we have other people that have orange toes. And both of those are indicators of COVID. And the fact that it's novel, what I'm talking about in a digital patient platform is going to be new. And we're going to have to pay a lot of attention to how we build in flexibility so that it evolves over time. We're in the startup process. And so in my thinking about this, and I think Deborah did a really good job of, you need to bring people from different points of view together, and you need to spend some time getting the current lay of the land. There have been people working on who continue to work on building physiological models. There are other people that are looking at doing digital twins for very small slices, either a virus or an individual. We need to bring people together to say, the only way we're going to get to where we want to get. So for example, testing a new vaccine against a virus, you can do that at a molecular level, really small, seeing does it target that, but it doesn't then take into account all the stuff that we were just talking about, when you get to the level of the full human or of the neighborhood, and behaviors in the neighborhood as well as individual behaviors affect the course of what's going on at a molecular level. And we need to be able to have a group that can help us think through how you build a model that is robust, that is practical, that's adaptable, isn't so complicated that we can't do it, but at the same time is more complicated than all of the models that are out there now. So Dr. Glazier, I think I'll stop there. I was right at the end of my time, I think, anyway. And we can proceed how you want.
Lovely, thank you. So we actually, despite the breadth of both talks, they were delivered with breathtaking speed. So we have time for some general questions before we go into our open forum. As usual, the request for the questions during the formal meeting time is that we try to hear from people who maybe don't speak up so much, and the questions should be kept relatively non-technical. And all gloves are off in the second session. So any questions for either of our speakers now?
I'd be happy to ask one to Debra Peters. I don't think this is a gloves off type of question. So, so great talk. I think the question is for the vector-borne disease and the modeling, how you would imagine expanding your approach to vector-borne human diseases like Zika, where you've got mosquitoes that you're concerned about. And maybe the further extension is if it's not necessarily vector-borne and you think about COVID. So the extensions, challenges and opportunities there.
Yeah, so we have a paper we just submitted on a West Nile human disease spread. So we've gone that direction. And then, Triple E, I think the issue is whether you have enough response data to actually work with. So Triple E, we think we have, we tried to do blue tongue, but we couldn't get enough data. So I think it's sort of, there's lots of environmental data, right? So when I think of the driver data, there's a lot of that. It's the response data that typically limits what we can do. So I think otherwise, we're open to certainly collaborating. Now we have the expertise in VSV, and we can sort of go to West Nile and Tripoli, but I think we would need collaborators, right? Really want to get with people who understand the system. And my group recognizes, my team here in Las Cruces recognizes, we don't know anything about these diseases. And so we need to have the experts. We listen really well. you know, and so then we can put it together, but we need the experts in the room. And I think we took 6 or 8 months just peppering the VSV team with lots and lots of questions, just over and over again. You know, we just asked them lots of questions about what are the vectors, where do they live, where do you see them? Is it different in New Mexico and Colorado or Montana? I mean, we have all these questions that before we actually, you know, went after the data. And I think people jump to the data a little too quickly, but we wanted to understand as much as we could before you know, it still costs me money to have my people get those data, right? So I wanted to be sure that I was going after the right data before we did. So then the second half of your question has to do with like COVID. So I think as long as there's a environmental aspect to it, right? I think we can expand this approach because I think it's sufficiently general, and that's sort of where our comfort zone is. So I think otherwise, if we wanted to go to other diseases that don't have any environmental, I'm not sure how I could help. I'm not sure I could be the best person to help, but I'd be willing to try. And so that I'll leave. You're ready to ask another one.
Couldn't one argue that all these emergent, zoonotic spillovers are environmental, or are you, the disruption of ecosystems? And I wonder, so it seems like you would be best positioned to be able to address these kinds of, predict potential for that.
Yeah, I mean, just I need people to work with because I'm not going to have that expertise, but absolutely, yeah.
Great, thank you.
We have time for 1 or 2 more general questions.
I have a question, perhaps for Debra Peters, and it's probably relevant also to Don Combs, which is the challenges of an interdisciplinary team. So maybe you could say something about how you went about putting the team together, recruiting, training, and communication, some advice, lessons learned, anything like that?
Yeah, so we had never worked together. So it wasn't like we had a history. Luis knew the people who were doing the right work, right? Like the insect people in Kansas that he knew about, but we had never worked together. So it was sort of starting from scratch. And I think identifying team players is really important. There are always going to be options on who to sort of bring into the team, but I think we were very careful to bring in people who wanted to collaborate, who were interested in listening to everybody's perspective and everybody's view. We don't have anybody sort of running over anybody else. I think also the team, everybody has sort of their own role. So that also helps, right? Because we acknowledge there are certain experts in things and they're the experts, right? So we go to them. I think we also have regular meetings. We actually meet twice a year. We have monthly phone calls, and then we were meeting in person. And we went around, we just kept moving. And we started in Fort Collins, and we came to Las Cruces, and then we went to Plum Island, and then to Manhattan, Kansas, and then we started around again, right? So we just had our 10th meeting. Now this one and the last one, of course, were virtual. But we actually went to each location, and that really helped a lot because each person could host it, had their own people. And I think the last thing I would say is that we've expanded. So we started with 5 or 6, and we're at 15, I think now. And so as we sort of, we're also, we start with one very general pattern, and now we're sort of looking, okay, how do the insects get across the border from Mexico? That's a separate question, right? So I think we're also expanding, our breadth of what our interests are, and so we need more people, and recognizing that when you need more people and bringing those people into the team.
Okay, there was a question for Donald in the chat. Could you imagine a version of the digital patient that was decentralized to avoid privacy worries about doing artificial intelligence on data being gathered and concentrated in a small number of places?
Yes, and thanks for bringing up one point as I was trying to cover a lot of ground that I think I only mentioned. Most of the physiological modeling that's been done so far is traditional. It's top down. It's based on the investigators' hypotheses and model and constructing it together. And that's a part of my looking for people to collaborate is I want to think about this in 2021, not in 1980, 97, 2005, where not only do we have top-down modeling, we have bottom-up modeling. And the point that you raise is an excellent one that is worthy of discussion and exploration. Can you park data in lots of different places and in real time quickly aggregate it to solve a particular problem? And that's kind of my notion, which I hope I covered in, is I think the digital patient is ultimately a confederation, not only of types of data, but of different models. And what we want to do is not be perfect, but be good enough. And that notion about decentralizing is a good observation. I want to just follow up to Debra's comment on the team. I've got a fair amount of experience in modeling and simulation, know people pretty well internationally as well. But this is, as I said, not a new effort, but a reinvigorated effort. And so we are going to start, in addition to myself and my co-principal investigator, with a staff of 4. One person who's totally dedicated to reading the literature and telling us what's going on, looking at the web. And that's the first person I've hired. And she's busily at work, going to educate us about what it means to be in 2021, because I don't know. We're going to recruit somebody who's a specialist in medical modeling and simulation, somebody who's a computational biologist, and somebody whose competence is in bioinformatics. And we think that team will give us the oomph to take these general concepts to the next level. We're recruiting now, and by the way, if it's okay, if it doesn't break a protocol, I would love to send an email to everybody who's on the list of this group, say if you know anybody who may be interested. I had originally thought about hiring PhD faculty types, but I've subsequently said I'm really interested maybe in postdocs or even really smart masters who may be more up to date than the other, but it also may be an opportunity for them to get involved. And so is that okay, Dr. Glazier, to send that?
That would be. I'm sure that everyone on the call and the working group who has graduate students and postdocs who are looking for positions would be delighted to know about the opportunity.
All right.
So we'd be happy to distribute them.
All right. We'll get it out tomorrow. Thank you.
Okay. Before we just come back to our usual slide to close the formal part of the meeting before we have our open discussion, which is that we always I would like your comments and feedback about how we can do better. We have some subgroup meetings scheduled, as you heard. We have some reports from subgroups. It's great. Let us know what you'd like to present. Any way we could publicize these Thursday meetings to reach a broader audience. As you see, our topics are diverse, and so we want to make sure that your communities are served by these meetings. Please let us know. Anybody who wants to have ideas for the steering group is most welcome to suggest them. And I think, especially in context of the 2 talks we've heard, we've been thinking a lot in the steering committee and elsewhere about the ultimate aims of this working group. And I think in many ways, the Digital Patient Project that we just heard about is very parallel to some of the things we've been thinking about. Anyway, we can translate the excitement of this group into actionable results is wonderful, and we'd like to hear your ideas. And so we'd appreciate anything that you want to talk to us about. Reinhard, do you have any subsequent things you want to talk about before we move to the open discussion?
Nothing.
James.
John, please.
John Rice. Yeah. Can I grab about 2 or 3 minutes here to introduce the idea that's been offered to us from IMAG?
Please, go ahead.
Okay. Bruce, if you throw that slide up. Basically, we got communication in the dissemination outreach group from Grace Pang that since their March meeting had been canceled the organizers prefer to wait till next year when they can do theirs live, that we've been offered the opportunity to create and host what would be the IMAG Multi-Scale Modeling Consortium annual meeting. And so having talked with my subgroup and talked with the steering group here, have had support for taking that on. And what I just want to quickly share is the kind of construct of it, that because it's going to be virtual, in spite of the limitations, like we would really rather see skin and eyeballs together, there's a lot of other things that we could do this time that would really help from the outreach perspective. So in working through some of these ideas, the conference, the way it's envisioned, and we have not, by the way, accepted the job yet. This is with you guys, because if you're not interested, we're not going to be able to do it. But I've envisioned it as having three primary parts. One is the typical programmatic IMAG, MSMC, their working groups, time for their working groups to report out, break out into breakout rooms for their working groups to have an hour meeting. For those of you who have been in Bethesda, you understand that part. The other part that's significant is the normal scientific meeting that the MSMC community has, which is the presentation of models and things and papers. But this, we have a lot more opportunity to reach out, because it's virtual, to people who normally don't get to attend the meetings in Bethesda. And so in this context, my kind of offer or request to you all is, I would like to set this up where the scientific part of this meeting is actually organized and hosted by the virus pandemic new working group, and taking advantage of all the topics that we have and the subgroups, that subgroup chairs would be willing to become the track chairs for a track of 2, 3, 4 sessions related to what your subgroup is trying to do, so that your own people can exchange information. But you can also do a call for papers, for virus-focused papers in your subgroup topic areas to construct your own tracks. And then the other part would be a thematic one. And just, right now, it's just to take advantage of the outreach things, to bring in people from other areas and other disciplines to do some sessions, some panels, interoperability panels on FDA and what FDA is expecting, panels from industry to hear from industry what they would like to be able to hire coming out of your university training programs, that type of thing. So with that much time available, I would ask if Jim, you could still assign me a breakout room that will not be open for at least 15 minutes. I would hang around and wait for people to finish up talking to speakers. And if you want to ask questions, otherwise we'll be following up with an email very soon asking you specifically if you want to talk about track sharing. Thank you.
Okay. Thank you, John. Okay, with that, our regular meeting is closed and we now have our discussion. We have two people with enormous expertise here. I hope that it will be a lively discussion. Feel free to unmute and ask any questions you like to either of our speakers. I have plenty, but I'll hold mine until the others have had their say.
I have one for Dr. Coombs. one issue in the digital patient is that I don't think I've ever heard anybody talk about it, is who owns the digital patient? Does the patient own the digital patient? Does the doctor own it? Does the doctor's hospital own it? Does the insurance company own it? Does the government own it? I mean, ownership defines where you have to start. And HIPAA says a patient owns their medical records, but in
Right.
practice, that's not true. In practice, patients have very little access to their records.
Right.
So can you give me some background? I'm sure others have thought about this, but what is your view of who would own a digital patient?
Well, again, first of all, we're really talking about, at least I am focused on the platform. And so if it's an open source platform that people, if they subscribe to certain conventions and want to import data, they can. Nobody's forcing it to. So the question, I think, really is who owns the data? And as you just said, in theory, the patient owns their data, but they don't own the data. For example, Norfolk has 15 databases about different aspects of the city that are neighborhood. And if they want to participate in this project by say, we want to build a profile for a certain neighborhood with certain demographic and income characteristics who are diabetic, then the diabetes information may come from state vital records, in which case it's public, or it may come from an aggregation of patients who opt in to share their data. But the ownership, at least, I don't want to sound grandiose at all because I'm not, it may not work out. But to me, the best analog of what I'm talking about is those folks who designed the World Wide Web in the early 90s. There weren't cell phones, there weren't email, there weren't apps, there weren't all of that stuff, but they built a platform that was robust enough you can hang all that stuff on it. And that's what I'd like to focus on because it's not there now. What we have now are an ongoing series of one-off studies about models. And the only way we're really going to move ahead to the whole thing is to say, we're going to think about the whole thing. I hope I didn't dance totally around your question.
No, that's an excellent answer. So one approach would be a web app that you can use your phone to get into and knows how to do things like I tell it who my doctor is and it knows how to contact my doctor and the doctor may refuse and all that kind of stuff. But yeah, that makes more sense than some software tool in a doctor's office because recent history has shown that digital medical records are terrible.
Yeah.
And every 5 years, doctor offices have to go out and buy a new one because the company that was making the old set goes out of business. So we need something more flexible. That's an excellent answer. Thank you very much.
I want to comment on this. This is important because this entire topic was actually raised in the integration subgroup and the reproducibility subgroup. There's a problem of licenses, and we just submitted a white paper for review there. The problem is not as simple as you can think, and people have different approaches towards approaching it. The problem is also with the data licenses. The problem is also with the software licenses. And when you start integrating things, you find out that even the things that seem obvious are far from obvious. I won't go into details here. So the question that Jim asked was very, very relevant. And your answer was actually something that I can allow me to express it in different words. You said, no, I'm not looking at the system, I'm looking at the spec, because the World Wide Web is actually a specification. You're looking at the standard. And if you're looking at the standard, I think Catherine Morse here, she will be talking next week. So you might as well make sure that you hear what she has to say, because even in the world of standards, there's an issue, elements of licenses and issues related to this.
Exactly.
But I want to move to a different topic, and actually the question for you is both to Debra and to you. You both, and this is a repeating question, I am asking it every week. I want to see your answers. You both use the term predictive. Do you really think you can predict? And how predictive are you? If you are already predictive, let us know. We want to know the first one who is really predictive. But If not, what do you really call predictive? Because there are other terms to use to figure out what will happen in the future, maybe forecast, maybe project. Predictive is wrong. We're telling wrong things to the people. Can we correct this? So this is my question to you both.
Deborah, you want to go first? You want me to?
Go ahead.
Well, I guess one part of my answer would be, if you remember the image I put up of data sources for the digital patient, that does not now exist. And if you don't have all of those different types of data, you can't truly be very predictive. You can be predictive about some big things. I mean, we know that. I mean, we know that if you are a heavy smoker, you have a statistically higher risk of a variety of things. And we've got data to show that. But can we predict at an individual level? No, because we don't have all of those circles of data. So that's my answer.
Yeah, and I appreciate that you're talking about other kinds of terms like, projecting and forecasting, because that's very commonly used to fudge it, right? To say, I don't really know, but I'm going to somehow get in the right quadrant, right?
Yeah.
So yeah, I mean, I think that's what we're trying to do. And our aim is to be predictive, but we also work with natural systems that are not amenable to prediction. So, we try to do the best we can. So, yeah, I appreciate your question, but it is really challenging, but that needs to be our aim, is to do the best we can. And if we have to put, uncertainty bars on it and probabilities on it, and if that's what you're asking, we can do that, certainly.
I want to say something. Jacob, you have to wait. Jacob, you get a lot of floor time. Let the other people speak a little bit. But I want to ask something to follow up on this then, which is, to me, both of you give a vision of integrated approaches to, we could say, forecasting rather than prediction. Although I actually am more interested in prediction because I do want to predict specific individual patient outcomes.
Right.
But to me, the platform concept is very powerful, because once you have predictions like the ones that Debra has, you can ask the question, how well did we do? And once you know how well you did or how poorly you did, you can ask the question, what is the missing mechanistic information or the missing data sets, if it's a data-driven model, that we could do to improve the quality of our prediction? You're able to bootstrap very quickly once you have something that does some forward prediction. It doesn't have to predict very far in time. It doesn't have to be perfect. But once you have something that you can shoot at, you have the ability to bootstrap. Weather forecasting is a good example of this. And so I think it's very interesting to think about what this technology allows you to do once you deploy it. The first versions of it don't have to be perfect to be useful. And I'm happy we could talk at some other time about whether we should say prediction or forecast. I'd like to give the opportunity to some of our other audience to ask some questions of our speakers. Jacob, you can come back in a bit. I see some people on the call. William said, I guess you can't talk. Is there anybody else? I see Jeffrey Morgan, Adam Knapp, Lorenzo. Do you have any comments or questions? Veronica, please, anybody? Anna.
Well, tonight is a little bit far of from my expertise, I must say. The only thing I can think about as a general question is what Debra has shown, if it can be scaled down to a cellular level, whereby the neighboring region which interacts, so according to certain parameters, are really cells, where is more my environment. I'm focused on single cell and neighborhood of cells. So it's a little bit generic. I understand that.
Yeah, so you're asking if the approach could be used at a very small spatial scale and very fast time scale. Yes, now I think the general concept, all we're really doing is we have local processes, and then we have processes that allow things to move in space and time, right? And so and focusing on the processes, is really important instead of just these correlation analyses. So I think in general, that sort of, conceptual model of that there are local processes, and when I can think of as spatial processes, I think that is highly scalable in both smaller and larger.
I think something that's important is that while this object process ontology for describing models is something that seems very natural once you do it, it's not something that mathematical biologists have in their bones. And so one of the things that to me was very valuable, I think that the analogy that Lorenzo drew between building a digital patient and building a model of the Southwestern United States actually have a lot of similarities. How you organize the subcomponents of the model, how you integrate divergent data sources, how you do whatever we call prediction or forecasting, and what we do with those results and divergences. Those, I suspect, are going to be the same, whether we're trying to build a model of the organ systems within an individual or of interactions on a large-scale spatial level. And so to me, actually, the combination today was very powerful because it showed the parallelism between trying to build a model of an individual digital patient and trying to build a model of the whole Southwest ecosystem of the United States. And so I maybe I'm hammering on that too much, but to me, the correlations and the similarities were more striking than the differences. Was there anybody else who had questions for the moment? I have another follow-up question, but I want to let other people speak first.
I've also got a follow-up if time permits.
Please, John.
This is for Debra again. So back in 2004, you had this nice PNAS paper on cross-scale interactions and their potential roles in catastrophic events and maybe counterintuitive ways that they impact catastrophe. I wonder since then, and given the experience you've had with some of these actual problems, to what extent this opinion is reinforced and to what extent you might be revising it and putting weight elsewhere. I'm just curious how your thinking has maybe changed since that time.
Yeah, so the value of bringing together, so that paper talked about desertification and fires, the Columbia space shuttle blowing up, a bridge collapse, right? And so I think that I think the power of that paper is comparing all of those very different systems and how those dynamics actually play out very similarly. So they start small, then they spread to the next scale, and then they spread there, and then they spread to the next scale, and it's very nonlinear, right? You can very much make a mistake by having a linear model, either above or below, right? And I think that so it also plays out for hurricanes, right? I mean, they start off the coast of Africa, come across. I mean, there are lots of actually processes that do that, and we can explain dynamics in that way. I guess what I also learned is that not everything does that. There are some things that are linear in between those, right? So I think that it actually has framed a lot of what I do. The Dust Bowl is another good example of a cross-scale interaction, very small scale. Every farmer was doing his own thing, his or her, it was all his at that time. They're all doing their own thing. But then the combination of those, a highly connected landscape that then led to the Dust Bowl. And then you can actually stop it, right? You can actually go in, if we had gone in, actually, well, the river did it, but we could have gone in at a particular point in time and said, we can't have this much connectivity in a landscape or it's gonna, take off. So I think it's very much sort of, it was a very early sort of thinking about cross-scale interactions, but it has led to much of what I've done since that time.
Great, thank you.
Other comments, questions?
So
I
have a
guess,
question
go ahead, Ruchira, please.
Yes, thank you. For Don Coombs, so this is perhaps due to the limitation of the format. So in the slide that introduced you, it referred to several other projects, and you also referred to projects in your overview of the history, such as BioGears and Muse and HumeMod and so forth. And at the same time, you're saying that you're starting a new effort. So perhaps due to the limitation of having both to give an overview of what is the effort and then also so there wasn't as much explanation of what distinguishes a new approach from all the approaches that are going on. Like it looks like human, for instance, is a current concern.
Well, so make sure you can hear me. Our focus is twofold. The first focus is to revisit the notion and flesh out of a platform, because nobody that would be able to be used by a variety of individual workers, researchers, so that Human might provide, they don't do, you couldn't use their physiological model to predict vaccine effectiveness, because we talked with them about whether they could do that. And they say, no, we don't do that. That's not what we're modeling. And so our notion isn't to compete with those who are already building models, whether those are viral models or community models, but rather to focus on how do we build a platform that allows us to reach this ultimate goal of why everybody's doing all the pieces. And so we're going to focus on that. We're not going to try to compete with Google or Microsoft or Amazon or anybody who's developing apps to do particular things, but rather to raise some of the issues that Dr. Glazier raised about what are the standards that people can agree to. It's going to have to be voluntary. We're not going to mandate this, but that's a long-winded answer to say we're trying to focus on the platform, on the middle, that enables all these pieces to come together if they only do certain things, define things the same way, have the data fields aligned, that sort of thing. So that's going to be our focus.
Focus on interoperability, you would say?
Yeah, I would say that. And then the other piece is to test that by developing a couple of models ourselves, again, where we have expertise, to see if the platform in fact allows us to pull all this data from different places and do better than what's out there now. Does that help?
Yes, thank you.
I guess I'd like to maybe follow up on that a little bit, maybe more to Debra to begin with, which is one problem you face certainly inside the body is that the boundaries of biological objects are fuzzy. There's a lot of boundary crossing, and the information that you have crossing boundaries is not always very clear. Physiology models make a particular kind of reduction of biology that may or may not be the most relevant for particular kinds of applications. And there's a lot of science we don't know about the proper way to define boundaries and information flow. And so how, in your modeling, you integrate a very large number of data sets. How did you decide essentially what the parameters were that you were going to be calculating and passing between your blocks, for example, in your models?
Yeah, so it's not actually it's not a simulation model. So what we were doing was it's maximum entropy. So it's actually a large scale multiple regression, basically. So we're not really passing information. We can get thresholds, right? And threshold sort of responses, like, the insects can't go beyond a certain temperature or precipitation. But what we were trying to do is we were actually starting from like nothing was known to generate hypotheses about what could be known, right? Could be happening. So I think that was actually a hypothesis generation exercise. But we were forming these relationships between sort of insect population dynamics and environmental variables that we could then use in that sort of correlation analysis.
Did you ever think about actually using all that structure that you built to build a dynamic mechanistic model? Looks to be like, even though you started out by doing correlation analysis of the data-centric model, you've in fact defined a lot of processes that could then be modeled dynamically as in an agent-based context, for example.
Absolutely, that would be fantastic. And so I've created numeric models for grass shrub interactions and desertification. And so it would be great to do that. I just haven't had the right person come in that could actually do that agent-based modeling for me. You're right, it'd be absolutely fantastic. That's the next step. Yeah.
Because it's really there that, to me, that the connection between the virtual patient concept inside the body and the federation of information and scales and your work would connect. Okay. Other comments? John, did you have something?
I have a question for Deborah, and are you at APHIS actually?
No.
But you work with them?
Yes.
Okay, and so maybe you can answer this, maybe not. But with regard to the COV2 organism, APHIS got involved in doing testing, and if I remember right, they've reported that they found the virus in 60 animals, non-human. What do they do with that? When, if I want to say, were there autopsies? Did they sacrifice those animals in order to do research to see, what is it about those animals that allowed them to actually become symptomatic? Did they become symptomatic? I can't find any information coming out of Department of Agriculture. And as a working group for virus pandemic, the scope of where we need to be able to go to learn about how this use case COV2 affects organisms, comparative immunology questions, like if it was horses but not cows, why?
Yeah, I know.
Okay.
Yeah, so they're not a research agency, right? So I work for the Agricultural Research Service. So we're the research agency within ARF, within USDA. So
Okay.
APHIS with us, they have the occurrence data, right? So the veterinarian calls them and says, look, we have a horse, it has lesions, it needs to be checked. APHIS has some labs, and they will then just document, is it VSV? Is it, something else? We don't know. And they just provide that information. So, and for us, it's all at the county level, which is useful. If you know about the West, our counties are huge. So we then work with an APHIS collaborator to get the actual locations. So that's the other thing. You sort of have to have sort of a connection to get the additional information that they can't post publicly.
I mean, I know people at APHIS that I've asked, and they didn't seem to know yet or something. Okay, never mind.
You got to find the right person, right? Yeah.
Yes. But the point here is we need to include that kind of data
Absolutely.
about pathogens.
Well, we would be nowhere without that APHIS collaborator with VSV, right? Yeah, absolutely.
Okay. John, you had
That's what our outreach group is for.
Jim, you had a question?
Send me an email and I'll ask my APHIS person if she can get you a name, because I think it really is finding the person who sort of led that, but they're just sort of documenting, right? They aren't researchers for the most part.
Okay. Time for one last question or comment.
I have a quick question.
Please, Reed, please go ahead.
Yes, I can turn my camera on, I suppose. So it's a real question for Dr. Peters. Sorry if I missed it because it's Thursday afternoon and I'm forgetting now, but the molecular virology aspect of this, like being able to track VSV by SNPs, I know there's some lineage work done. What's the current status of that? And is it sufficient to use to validate your forecasting of when things will come? Can you track specific lineages over time? Is that improved, or is that a gap? Or is that not? Or is
Right. That's what Luis's lab is doing.
Yeah.
I think that they, I'm going to be very limited in what I can tell you, but I think that they can at least track the lineages that it came from a certain lineage in Mexico, and then in different parts of the country in the U.S. are different lineages. So I think that, but that's his role. So
okay.
Yeah.
Okay. Thanks.
Okay, I wish we had more time, but I think we're out of time. We really appreciate our speakers being willing to stay on. I hope that the discussion was productive. I can see there's going to be a lot of opportunity to ask more questions, and I hope that this leads to integration of projects and ideas. I guess, Deborah, I was going to close with one question, which is, if you think about, if you had the equivalent of a digital patient for all of your horses and all of your insects, would that be, at the moment, because you have a data model, that might not be so useful. If you were going to go to the sort of mechanistic dynamic model, how would you use it if you had it?
So that would reduce our error on our estimates, right? Because right now we have quite a bit of error when we do sort of our regressions, and I think that having that additional knowledge would reduce that error.
That's a cool question. What if you had it for even just one? What's the simplest organism we could imagine that we could actually set out to build a digital version? That's cool.
Okay, well, thank you again, Deborah. Thank you, Don. We really appreciate it. I hope we'll be hearing from both of you again. And I hope that your presentations will lead to lots of emails and lots of offline discussion. Thank you again. I appreciate
Thank you
everyone
for the
sticking
invitation.
around.
And you will save and send out the chat to everybody, please. Thank you.
Okay, with that, I'll call the meeting to a close. Thank you again.
Bye.
See you next week.